We are still busy looking at the food microbiology and public health. So far, we have looked at incidences of foodborne illnesses. We have also compared the different recording systems for occurrence of foodborne illnesses in different countries. We focus on the limitations of such systems. We also discuss the consequences of foodborne illnesses as well as the occurrence of emerging foodborne pathogens and factors contributing to development of such pathogens. We end this theme by discussing the alimentary tract or the gastrointestinal tract as the site at which foodborne illnesses occur. And then we look at the types of foodborne diseases and their disease mechanisms. The alimentary tract or the gastrointestinal tract is involved in digestion and absorption of food. Most foodborne illnesses cause a dysfunction in the alimentary tract, resulting in symptoms such as diarrhea and vomiting. However, in addition to the involvement of the alimentary tract in digestion and absorption of food, it is also a habitat to microorganisms and a route by which they penetrate the body. You are advised to revise this section that explains what happens to the foods in different parts of the alimentary tract. We are focusing here on the microorganisms residing within the gastrointestinal tract and their roles. The gut contains more than 400 species of bacteria, which are estimated to be more than the numbers of the cells in the body itself. The numbers of these bacteria increases the further you move down the gastrointestinal tract, with a decrease in the flow rate of the food material through the tract. Now, the higher the flow rate, especially in the duodenum, surpasses the rate at which microorganisms can multiply. And at this high flow rate, only those microorganisms that are able to adhere to the epithelial cells can persist. If we look in terms of numbers at the microorganisms that are present in the different sections of the gastrointestinal tract, we will see that what is noticed is that within the mouth, the numbers are way lower, and that is also due to the presence of antimicrobial compounds such as the lactoferrins and the lysozymes. If you go to the further down the gastrointestinal tract, the stomach, which contains gastric acid, has also lower numbers, and this is due to the acidity of the gastric fluid that is secreted within the stomach. In the small intestine, the different sections as well contains different numbers, with the duodenum containing between 10 to exponent 2 to 10 to exponent 3 microorganisms per mil, the jejunum having between 1,000 and 10,000 per mil, and the most populated part of the small intestine being the ileum, which contains about 1 million cells per mil. The types of microorganisms that are present in the healthy individuals is mostly in the small intestine. It is comprising mainly of the lactobacilli and the streptococci. If you look at the large intestine, which is the most densely populated in terms of microbial numbers, it contains mainly the Bacteroides as well as the Bifidobacterium. These two comprises about 99% of the population, but it has also a presence of other microorganisms such as your enterococci, your lactobacilli, yeast, staphylococcus, pseudomonas, and so forth. Now, these microorganisms in the large intestine, they constitute between 25 to 30% of fecal material, and in total they contain about 10 to exponent 10 to 10 to exponent 11 colony forming units per gram of the fecal material. The rest of the fecal material contains indigestible food components, the epithelial cells that are shed, some minerals, as well as bile. The gastrointestinal microflora that we just explained has a very critical role to the host cells or to the individuals, as the overall health of an individual will be impacted by the conditions of the gastrointestinal microflora. So the interaction between these microorganisms and its host can have both positive and negative aspects. However, the overall health is very much dependent on the integrity of the microflora, where there is a balance between the beneficial and the detrimental effects. What that means is that in a healthy individual, there has to be more beneficial microorganisms as opposed to those ones that are resulting in diseases or those ones that have detrimental effects. The role of the gut microflora in protecting the host against infection has been established since it has been observed that absence of the normal gut microflora will allow colonization of the gut by pathogens such as Clostridium difficile, which when the normal microflora has got beneficial microorganism, inhibit this particular pathogen from growing and producing the toxin, which will result in the disease. Furthermore, it has also been observed that the infective dose of some of the pathogens is reduced when the gut microflora is disturbed. Now, the mechanisms by which the gut microflora offers protection to the host is due to a number of mechanisms, and firstly it could be due to occupation of the attachments on the epithelial side by these beneficial microbes, which are part of the normal microflora. It can also due to production of antagonistic agents such as the organic acids and the bacteriocins, which have got inhibitory properties against pathogenic microorganisms, and also can be due to stimulation of the host immune system, whereby it has been established that when there is a balance in the normal gut microflora, there is an increase in the amount of the circulatory antibodies. So the major physiological functions of the gut microflora can include both the barrier function, modulation of the immune system, production of nutrients, as well as involvement in their digestion and absorption. And furthermore, they can also stimulate the bowel motility. So this indicates that the normal gastrointestinal microflora is very beneficial to the host. Unfortunately, despite the importance of the balance between the beneficial and the detrimental members of the gastrointestinal microflora, it is unfortunate that it is not always the case that the balance will be maintained. There can be factors that will perturb or disturb the balance, which changes the gut microflora to a situation whereby there are high levels of the detrimental microorganisms as opposed to the beneficial ones. A number of factors can result in the disturbance of the normal gut microflora, and based on what has been mentioned, you can see that this is something that cannot be avoided. Some of the factors that result in the disturbance of the balance are lack of food and water, traveling, which exposes us to different conditions, use of antibiotics and drugs for treatment of diseases. We know that antibiotics have got antimicrobial properties, so among killing those microorganisms that results in infection, they can also kill the members of the beneficial microflora. There's other factors such as emotional stress, poor diet, as I've already mentioned, lack of food and water as well. And also, as we get older, the balance becomes disturbed. So this loss of indigenous microflora, which could be due to any of the factors that I've mentioned, can result in a predominance of the microflora by harmful bacteria, and this is due to the availability of the attachment sites or adhesion sites of the intestinal epithelium, which were previously occupied by the beneficial microorganisms. So as soon as those microorganisms die, for example, when they are exposed to antibiotics, the sites to which they were attached become available, and the pathogens that are higher in numbers now are able to bind to those. And unfortunately, in most cases, the consequences of that is that the people will now become susceptible to the infections. Dominance of the epithelial sites by the pathogenic microorganisms predisposes the individual to foodborne illnesses. Although most of the foodborne illnesses will result in symptoms which are confined to the gastrointestinal tract, there are also those foodborne illnesses such as botulism, listeriosis, as well as typhoid fever, which will also affect sites that are far away from the gut, although the initial entrance of the microorganism or the causative agent is through ingestion of food. So even in those cases, for example, botulism, which affect the nervous system, or listeriosis, which can result in the pathogen getting to the placenta and causing abortions and so forth, the alimentary tract or the gastrointestinal tract still serves as that site of entry by which the pathogen gets access to the human or to the host. And most of those pathogens that are confined to the gastrointestinal tract will cause diarrhea or vomiting as common symptoms indicating illnesses. Since diarrhea is a common symptom, we are going to discuss it in some detail. Diarrhea is excessive evacuation of the feces that are too fluid, which is a result of the inability of the gastrointestinal tract or of the gut to absorb most of the water it receives. And it can also be to increase secretion of water into the intestinal lumen. Ability of a pathogen to cause disease could be as a result of its ability to damage the host. And damage to the host can be due to production of virulence factors such as toxins, but can also be to the ability of the pathogen to adhere to the mucosal surfaces, secrete proteins, and invade host cells. However, toxins are frequently a direct cause of diarrhea, and there are different types of toxins. The different types of toxins that exist include the exotoxins, which are released extracellularly by the organisms, or they could be the endotoxins, which are the fever-producing lipopolysaccharides, which are released by bacterial lysis of the outer membrane of the gram-negative cells. There are different types of exotoxins, and these are grouped based on their impact or their effect on the host cells. There are enterotoxins, which act on intestinal mucosa, causing diarrhea. There are cytotoxins, which kill host cells and host tissues. And there are also neurotoxins, which interfere with the normal nervous transmission systems. Whether a foodborne pathogen causes a disease through production of any of the different types of the exotoxins that we have explained, or through production of endotoxins, or through its ability to bind to the mucosal surfaces, the types of foodborne diseases can be divided into two broad categories, namely foodborne infections and foodborne intoxication. Foodborne infection refers to those cases where the pathogen itself, so the viable cells of the pathogens, are ingested together with the food, and that food enters the gastrointestinal tract with the pathogen, and the pathogen is able to multiply within the gastrointestinal tract and start causing disease. On the other hand, foodborne intoxication, the food that is ingested may not contain the viable cells of the pathogen. However, what is critical is the presence of a toxin. So before consumption or before processing of the food, the pathogen need to have been present in the food product and was able to grow and produce a toxin which survive within the food product until that particular food product is ingested. So even though some further processing of the food product can kill the microorganism, if it leaves the toxin intact, it is that particular toxin that is going to cause the disease, even though the producing organism itself is no longer present in the food product or it's dead. So that is the two broad definitions or broad descriptions of the different types of foodborne diseases that can exist. The types of foodborne diseases or foodborne poisoning are sometimes divided into three, and this division indicates that the foodborne infections can be divided into two types. So we will have foodborne intoxication, which is the ingestion of a preformed toxin. But in case of the foodborne infection, it can either be a non-invasive infection or an invasive infection. Let's explain the three types of food poisoning. The foodborne intoxication or the ingestion of a preformed toxin, toxins of the pathogens such as Staphylococcus aureus, Bacillus cereus, Clostridium botulinum, in rare cases the ingestion of the toxin produced by Clostridium perfringens, are the ones that result in the disease. And this kind of diseases or poisoning are characterized by a very short incubation time. So the period between the ingestion of a food which contains the toxin and the onset of symptoms is very short. The non-invasive infection is when viable bacteria ingested with food colonize or grow within the small intestine. It is severe in cases where there is disturbance to the normal microflora, gut microflora, as we have explained the importance of the normal gut microflora. Now, in those cases, the pathogen adhere to the epithelial surface by binding to the microvilli through production of adhesins. After the pathogen attachment, it produces an enterotoxin which changes the flow of electrolytes and water from that of absorption to of secretion. So it stimulates more secretion of electrolytes and water, while at the same time there is inhibition to absorption, or there is reduced capability of the system to absorb those electrolytes and water. And the mostly studied example of a non-invasive infection is that caused by Vibrio cholerae, and we'll discuss in detail how Vibrio cholerae enterotoxin causes diarrhea. The third example, which is the second type of a foodborne infection, is invasive infection. In this case, the pathogen invade and pass through the epithelial cells to multiply in the lamina propria, and this is the connective tissue underlying the enterocytes or the epithelial cells. The exact mechanism by which invasive infection occurs is unknown, and there is a number of suggestions or a number of possible mechanisms that have been observed in different pathogens. For example, in Salmonella, there has been observed that there is a production of a heat labile enterotoxin as well as a cytotoxin. In Shigella and enterotoxigenic E. coli, the cells invade the colonic mucosa producing cytotoxins, and in some cases for Shigella, there is production of enterotoxins. So foodborne infections can be either invasive or non-invasive, depending on whether the pathogen is confined to the intestinal lumen, which is non-invasive infection, or whether the pathogen multiply in the lamina propria, which is now invasive infection. Since non-invasive infection caused by Vibrio cholerae is the one that has been widely studied. We conclude this section by discussing the mechanism of Vibrio cholerae infection in detail. Now, Vibrio cholerae, after its ingestion with contaminated food or water, binds to the enterocytes or the epithelial cells and produces an enterotoxin. This is referred to here as the cholera toxin. So what that toxin does is that it will bind to the enterocyte. This toxin is made up of 5 B subunits as well as 1 A subunit indicated in black. So the 5 white subunits are the B subunit, while the black one is the A subunit. The B subunit is the one that binds to the receptors on the enterocyte, and after binding they create a hydrophilic channel in the membrane through which the A subunit can pass and enter the cells. The A subunit, as indicated, will now enter the cells, so it will traverse through the channel created by the B subunit. Once it is inside the cells, it transfers the ADP ribosyl group derived from cellular NAD to a protein which regulates the activity of adenyl cyclase. So it transferred that ADP ribosyl group to the G protein which regulates the activity of adenyl cyclase. Usually, activation of adenyl cyclase by the G protein is reversible. So when there is high levels of cyclic AMP which is produced by adenyl cyclase, then it will be inactivated. However, the transfer of the ADP ribosyl group to the G protein, which regulates adenyl cyclase, locks the enzyme into its active sites, leading to accumulation of cyclic AMP. So what that means is that there is irreversible activation of adenyl cyclase, which means the adenyl cyclase will remain locked in its active state, leading to production of a lot of cyclic AMP. Because it is locked in that active site or in the active state, there will be accumulation of cyclic AMP. Accumulation of cyclic AMP inhibits absorption of the sodium and chloride ions, while at the same time it stimulates the secretion of the chloride, the bicarbonate, as well as the sodium ions. So that indicates that there will be accumulation of a lot of ions within the system or within the lumen. In an attempt to maintain osmotic balance, the transfer of electrolytes is accompanied by massive outflow of water into the intestinal lumen. So the system is trying to balance the accumulation of the ions that are now secreted into the lumen. The outflow of water, however, is so much or is too much that it exceeds the absorptive capacity of the large intestine. That is, the amount of water that is secreted is way too much for it to be absorbed by the intestine. And since it cannot be absorbed by the large intestine, it is passed with the feces, which is why there is a resultant too fluid feces, which is referred to as the diarrhea. So this is generally the mechanism of action of the Vibrio cholerae enterotoxin, which leads to locking of the enzyme adenylate cyclase into the active state, resulting in accumulation of cyclic AMP. And the consequences of accumulation of cyclic AMP is inhibition of absorption of the ions, stimulation of secretion of the ions at the same time, and then that results in massive outflow of water into the large intestine, which exceeds the ability of the large intestine to absorb the water, and that is what results in diarrhea. Most of the foodborne pathogens that are non-invasive and that produces enterotoxins, though some could be due to not activation of cyclic AMP, they can use the similar kind of mechanism. So the mechanism of Vibrio cholerae is the one that we will use as a model system to demonstrate how the non-invasive infection occurs or results to diarrhea in the infected individuals. This now brings us to the end of our discussion of the general features of foodborne diseases, and it takes us to the discussion of bacterial as well as non-bacterial agents of diseases. So we are going to look into detail at the different bacteria or viruses or protozoa that causes foodborne illnesses, looking at their specific virulence factors, their specific symptoms of disease, and so forth.